In this lesson, we'll continue our discussion of muscle tissue by taking a look at a complete contraction cycle. We started it in other lessons, but now we're going to take a look at the whole cycle. And then once the muscle fiber is done contracting, we'll take a look at how it relaxes. So we can think of the contraction cycle as a series or sequence of events that ultimately cause muscle contraction. The first two dots here we actually did already. In the previous lesson, we saw how the active site was exposed. If you remember, once calcium was released, it bound to the troponin. Troponin changed its shape and pulled the tropomyosin off of the G-actin, exposing the active sites. Right, so that we did already. Then once the active site is exposed, the thick filament, the myosin head, will jump up and bind to the actin, forming a cross bridge. Right? So these two dots we did. So in this lesson, we want to take it a step further. We want to see once the cross bridge is formed, we're going to see that the myosin head is going to pivot, forming a power stroke. Then it's going to detach from the actin and do it all over again, right? So we actually have to reactivate the myosin. In these last three dots, we're actually going to see the role of ATP, right, which is our form of energy to do muscle contraction. So ATP is going to become important in the last three dots. So here's an overview from your book, just kind of showing the steps, how step one leads to step two, leads to step three. I want to go through each one of these individually. So just to kind of reorient what we are, here in the middle we have a strand of actin, right, F-actin, and you can see the tropomyosin, that's this kind of turquoise braided strand right here, covering the active sites. That means the muscle fiber is relaxed. Here's the troponin. Right over here they have it labeled, right, the troponin. Then above it is a strand of thick filament. This would be myosin. And then a thick filament below it. This would be myosin also. And then we can see the myosin heads here. Okay. So this myosin head, let's take a look at this one here, is actually what we call in the cocked position. It kind of reminds me if you took a pistol and you took your thumb onto the hammer and you pulled it back. Now it's ready to fire, right? If you pull the trigger, it's ready to fire. This is in the cocked position because ATP, adenosine triphosphate, was actually split into an ADP and a phosphate. When we split ATP into ADP plus phosphate, that releases energy, and that energy was used to put this into the cocked position. Okay, you can see here that calcium now is starting to become present in the sarcomere, in the area of the sarcomere. Let's go to the second step. Once the calcium is present, calcium is going to bind to the troponin. The troponin changes its shape and pulls the tropomyosin away. Here's the tropomyosin. See, now it's pulled away, and we can see the active sites being exposed. Right here, we get a really good look at the active sites. Once those active sites are exposed, the energized myosin head is going to jump up. Okay, so remember we said before this was in the kind of in the cocked position. It will jump up once these active sites are exposed. Right, so now we have this attached. This is what we call the cross bridge. Right, this is going to be the cross bridge. Once that cross bridge is formed and there's an attachment, the myosin head, if you notice, I'm going to go back a slide, will actually pivot. Remember, this is a hinge right here. So this is actually going to pivot. And now the whole actin is going to get pulled over a little bit. Okay, so I'm going to go to the next slide. Watch this. And you see the myosin head pivots and it now moved the actin. Here's the arrow showing it's moving it towards the center of the sarcomere. Once this pivoting happens, the adenosine diphosphate and the phosphate molecule are thrown off, right? We can recycle this ADP to make ATP. I'll show you that in future lessons. So now what we have to do is we have this myosin head attached to the actin. We have to detach it. Watch the next step. A new ATP jumps onto the myosin head, and that causes detachment. This ATP is going to split and release energy. Watch the next slide. It splits into an ADP and a phosphate, and notice this whole myosin is now pulled back into the cocked position. Watch this, right? So I went back again. So here was the detachment. That happened when the new ATP jumped on the myosin head. Once this ATP splits and it releases energy, we're going to pull this whole thing like the hammer of a gun, like a pistol, right? The hammer of a pistol, pull it back like with our thumb into the now cocked position. And once these are exposed again, this can jump back up. Okay, and this continues to happen. So now again, it's in the charged position. Okay, so those are the steps. Again, I'm going to go through it one more time just because it's a little bit complex. Right, here's the muscle at rest. How do I know that? The tropomyosin is covering the active sites. I have a charged myosin head. It's in the cocked position. This happened from the last previous muscle contraction where ATP was split into ADP and phosphate. That release of energy put this into the cocked position. Now we see calcium present. Right? Once calcium comes in, it's going to bind to the troponin. Troponin changes its shape, pulls the tropomyosin away, exposing the active sites. Once the active sites are exposed, this is going to jump up, forming a cross bridge. Right? So that's our next step right here. We have the cross bridge. That's important. Once the cross bridge happens, this myosin head is going to pivot. We call that a swivel. It's going to pivot, and now, since it's attached to the actin, it moved the actin over a little bit. Once it's swiveled, right, once it pivoted, the ADP is thrown off, right? The ADP and that 1-phosphate is thrown off. That causes that's just thrown off. So now this makes this available for a new ATP to jump on. That causes detachment. Once this new ATP jumps on, that causes detachment. This is going to split into ADP and phosphate, and that cocks this into the ready position for this to happen all over again. All right, and that's the contraction cycle. And now you can see the importance of ATP. So as long as we have ATP, we can produce energy, we can continue to do muscle contraction. Right? Once a muscle can't produce ATP anymore, and we'll talk about this in a later lesson, a muscle actually becomes fatigued. So, taking that information, when a muscle cell contracts, it produces tension, right? That produces like a pull on our bones to produce movement. But we got to produce, and I'm going to go into this in the next lesson, we have to produce enough tension to overcome the load, which is the resistance, or something maybe that we want to lift, or just even the weight of our body. Now, the entire muscle shortens at the same rate because all of the sarcomeres in a muscle fiber are contracting together. Right? And again, I'm going to show you this in more detail in future lessons. Also, the speed of the shortening depends on the cycling rate, right? How the number of power strokes that are happening per second. So the more power strokes that are happening per second, the faster the muscle is going to contract. Again, we're going to look at that in future lessons in more detail. Now, a muscle contraction, right, how long it will contract, it depends on a few things. It depends on how long the nervous system is stimulating the muscle at the neuromuscular junction, the presence of free calcium, right? If we don't have enough calcium, the muscle fiber is not going to contract, so we need to have adequate calcium, and we also need plenty of ATP. That means we have to have things like sugar and glycogen to make the ATP. Right? So these three factors are important for determining how long a muscle contracts. Nervous system stimulation, the amount of calcium in the muscle fiber, and then how much ATP we have. So now that we know how a muscle fiber contracts, what happens to make a muscle fiber relax? So once the nervous system stops stimulating that muscle fiber, the calcium that was in around the sarcomere is going to be pumped back into the sarcoplasmic reticulum. As the amount of calcium goes down around the sarcomere, right, in the cytosol, calcium is going to detach from the troponin. Then the troponin changes its shape again, but back to its original shape, pushing the tropomyosin over the active sites, right? The tropomyosin is going to be covering the active sites. If the tropomyosin is covering the active sites, then the myosin heads can't form the cross bridges and we don't get contraction. So kind of the everything kind of happens in the opposite. Calcium levels go down, the troponin reverses its shape, pushing the tropomyosin to cover the active sites again. Kind of happens in reverse. So this is just a slide showing, again, everything we went over to cause contraction, just kind of a review, things that initiate a muscle contraction. I'll go over this again. So here's the electrical impulse coming down the neuron. It comes to the axon terminal, which is the end of the neuron. Then it causes the release of the acetylcholine here by exocytosis, right, from a previous lesson. It diffuses across the synaptic cleft. It binds to the acetylcholine receptors. That stimulates now a new action potential on the muscle, right? So we just moved the red line from the neuron to here. The action potential travels down the T-tubule. Once it comes down the T-tubule, it stimulates calcium release from the SR, the sarcoplasmic reticulum. Once the calcium is released, it binds to troponin. It pulls away the tropomyosin, exposing the active sites, and now the cross bridge can happen. And once the cross bridge happens, the myosin head will swivel, pulling the actin right here towards the center of the sarcomere. Right? So all these actins are pulled in the center of the sarcomere. And on a previous lesson, we had called that the sliding filament theory, right? The actin slides over the myosin. Okay, so this is a good kind of summary. It doesn't have a lot of detail on it, but kind of a summary of what happens. So what happens on the reverse is that once the nervous system stops firing, notice, right, no more acetylcholine in the synapse, right, the acetylcholinesterase broke it down. No more electrical signal here. No more electrical signal going down the T-tubule. That means calcium is not going to be released anymore. But what's going to happen is calcium is going to move back into the SR to be stored. If the calcium is in the SR, that means the troponin reversed its shape, put the tropomyosin back over the active sites. If the tropomyosin is covering the active sites, the myosin heads can't bind, and we don't get muscle contraction. And watch what happens. The actin slides back, and now the sarcomere lengthens. Right? So this is what's happening in relaxation. It's the exact reverse. Okay, the last slide. Just a little bit about rigor mortis. So people know that after death, there is an actual period of time where there's a fixed muscular contraction. The reason this happens is, guys, the muscles, when somebody dies, the ATP runs out, right? The muscle doesn't have a lot of ATP in it, and the ion pump ceases to function where we pump the calcium back into the SR. So the calcium stays built up in the cytosol, and that produces the contraction of rigor mortis. All right, eventually that will go away also. Right, so that concludes this lesson on the contraction cycle and relaxation.